Classical Darwinian selection focused on what is now called the phenotype, specifically the fully developed structures and functions that organize the tissues and organs and shape and form of the individual in ways that facilitate survival and reproduction in particular niches or ways of life. In Chapter 14, I argued that this should be called the ecological phenotype to distinguish it from developmental toolkits, which also evolve, which I called the generative phenotype. A general trend in evolutionary theory that has taken place since Darwin has been the expansion of evolutionary theory with an increasingly detailed, empirically based understanding of the inner workings of interacting cells. None of this invalidates the core framework of metaphorical trial and error that Darwin reported in the first edition of On the Origin of Species in 1859, but we have seen profound changes in our understanding of hereditary variation. For plants and animals, however, the most important way that variation in ecological phenotypes is manifested is through genetic determinants that control the process of development. Development generates biological organization. In his introduction to The Making of a Fly, Peter Lawrence puts it this way. In my opinion, the heart of development is the step-by-step allocation of cells to more and more precisely determined fates. These allocations have to occur in the right part of the embryo. This key process can be broken down into two questions. How are the cells chosen by position? What happens inside a cell when it becomes allocated to a specific fate? That is, what are the molecular and genetic changes? A good deal of progress has been made in answering these questions. From cytoplasmic determinants in a single cell to specialized lineages of interacting cells. Focusing on animals, it is convenient to begin our analysis of development in each generation with the fertilized egg. However, a critical aspect of development takes place at an earlier time, at a time when the eggs are being produced by the mother. Cytoplasmic determinants in the egg are primarily encoded by the maternal genome. These generative phenotypes are known as maternal effects. They include a variety of RNA and protein molecules, including transcription factors and morphogens. Bicoid was the first protein identified as a morphogen, and is expressed early in the development of the Drosophila embryo. Bicoid is a morphogen because it is expressed as a gradient along the long axis of the egg and plays a major role in determining the various regions along the anterior-posterior axis of the developing embryo. Bicoid acts as a transcription factor that binds both RNA and DNA targets, using its homeodomain to regulate their transcription and translation, respectively. Bicoid is one of the most important discoveries made by Nussel and Volhard and colleagues during their Nobel Prize-winning analysis of mutations that are lethal to embryos and had therefore been previously missed. Development of techniques that made it possible to visualize the bicoid protein gradient in developing embryos is one important example of the ways that advances in empirical techniques have advanced our understanding of both development and evolution. The bicoid protein drives the differentiation of embryonic cells along the anterior-posterior axis, and its homeodomain and nucleic acid targets exhibit the deep homology that is one of the most remarkable general principles of evo-devo. More recently, other factors that synergize with bicoid have been described. Cytoplasmic DNA and cleavage. More recently, a powerful gene editing technology known as CRISPR has made it possible to trace the entire lineage of the mouse embryo. Briefly, the technique creates a sort of genetic barcode that allows investigators to trace the specific patterns of cell division and differentiation that give rise to a multitude of highly specialized cells during embryonic development. Gastrulation. As implied by the tongue-in-cheek quotation by Lewis Wolpert at the beginning of Chapter 11, gastrulation marks an important milestone in life, not only for us humans, but for the rest of the animal kingdom as well. For both vertebrates and invertebrates with a central nervous system and an anterior-posterior axis of development, gastrulation establishes the general tubes-within-a-tube architecture of the individual. In vertebrates, one of those tubes is the brain and spinal cord, while the other consists of the organs of the digestive system. Formation of the body plan and organs. Long before the advent of molecular genetics and evo-devo, construction of fate maps, along with detailed step-by-step descriptions of the developing tissues of the embryo, were a principal activity for embryologists both in the laboratory and in the classroom. Gastrulation brings particular layers of embryonic cells, known as the germ layers, into proximity, where they communicate extensively in ways that coordinate the gene expression of neighborhoods of cells to produce complex shapes, forms, and functional capabilities in tissues and organs. One of the rites of passage of pre-medical students that continues to this day is to learn the highly detailed anatomy and physiology of developing embryos, not only of human beings, but also of other vertebrates that can be studied in the laboratory. As an undergraduate student at UC Berkeley, I have vivid memories of the embryology lectures presented by Richard Eakin. In his well-attended presentations in one of Berkeley's largest lecture halls at the time, Professor Eakin would astound hundreds of us as he would draw, entirely from memory, the most beautiful and detailed pictures describing the various stages of embryogenesis, using a different color of chalk for each of the 3 germ layers, beginning with the fertilized egg and then proceeding through gastrulation to organogenesis. Professor Eakin used blue for developing tissues and organs derived from ectoderm, red for mesoderm, and yellow for endoderm.